Hi, everyone. Welcome to our next session. Our apologies for the slight delay.
Hello, everyone.
Okay, Maurizio.
Yes, Andrea, give me the okay when I can start.
Yeah, ready.
Terrific, thank you. Good morning, everyone. This is Maurizio De Pita speaking and sharing the session on synaptic physiology and introducing the keynote speaker of this session, Yulia Dembitskaya from the Collège de France in Paris and the Institute of Bioorganic Science of the Russian Academy of Science in Moscow. But Russia, of course. Yulia is based currently in Paris. So it's really a pleasure to introduce a dear friend of mine. Yulia and I share several passions, and predominantly the one for astrocytes and neuroglia interactions, along the one for wine and very good food, preferably Italian one, though not French one. That's a bias for me against the use of butter instead of olive oil. Anyways, I would like to just briefly mention the exceptional background of Yulia. Yulia is an experimentalist. I think it's a master of many techniques. She is specialized in two-photon calcium imaging, electrophysiology, and a lot of other approaches that she implements in studying not only neuroglia interactions, but also extracellular matrix and extracellular space and volume signaling. She has done her training in Russia, and then she moved to do her PhD with Alexei Semyanov in Okinawa. At the Okinawa.
No, it was actually.
It was in Russia. Okay, sorry.
In Riken. No, it was in Riken.
It was in Riken. Okay, sorry, my bad. Then she moved to Alexei Semyanov. No, I think you were in Alexei Semyanov lab in Okinawa. Then she moved back to Russia because she can't really avoid to leave her mother Russia. And then she moved to Paris, where she is gladly entertaining herself for the past 4 or 5 years, I believe, and accomplishing her postdoctoral training at Collège de France in the lab of Laurent Venance, and they're collaborating with many other local investigators, along with Michael Graupner and Pierre Magistretti. So welcome, Yulia, and I leave the stage. You have more or less 45 minutes, after which the question and answer session. Thank you.
Thank you so much, Maurizio. It's a great introduction. And hello, everyone. Today I will tell you about my project that I did at Collège de France, as Maurizio said, about lactate supply that overtakes glucose when neuronal computational and cognitive load scales up. Just a moment, I will move to the next slide. Okay. So everyone knows that our brain is very expensive organ. It represents only 2% of our body mass, but it consumes about 25% of all glucose and 20% of oxygen. So it's enormous for this size, and it has its reasons. So in recent studies, it has been shown that functional mapping studies, like using fMRI and the measure of blood flow, that metabolic demand of the brain increases when the neuronal activity and increases. So it correlates with blood delivery and oxygen utilization and glucose utilization. And glucose is the main source of energy in the brain. It's all well known and can be oxidated by different processes and used in different metabolic cascades to provide energy for the brain. Also, like, it can be oxidative or non-oxidative, and glycolytic metabolism increases with corresponding elevations in brain activity. Oh, something happened with the presentation.
Was it? Sorry, it's fine. Was it working while you
were?
Yes. Okay, so it might be a load of memory. Sorry about that. So Maybe you want to restart it again, and
Yeah, I'll do that.
okay.
Can you see it now? Well,
Yes, we can see.
okay, perfect. Well, so
Go ahead.
here's one of the schemes that's showing the glucose metabolism in the brain. There are three major cascades that are responsible for generation of energy. First, it's glycolysis, where glucose degrades to pyruvate and lactate. Part of it goes to mitochondria to 3-carboxylic acid cycle, and part of it goes with oxidative phosphorylation to generate ATP. The other important cascade is pentose phosphate pathway that also generates energy, mostly in the form of NADPH. And the latter, it's glycogen in astrocytes stored that is unique feature of astrocyte, but it's very important source of energy. Okay, I think I have it. This problem with the slides again. Sorry. Okay, astrocytes, it's a very important player of brain energy metabolism. They are able to feel the neuronal activity and respond to this. For example, when neurons release glutamate, astrocytes start actively glycosis. And when synaptic activity is high and there is a potassium accumulation in extrasynaptic cleft, also glycogenolysis starts in astrocytes. So basically, astrocytes on demand produce energy for neuronal activity. So to regulate the supply of energy, astrocytes, they have several cascades that control the blood flow in the brain. So some of them are increasing the blood flow, some of them are decreasing. And this is also dependent on neuronal activity. When glutamate release and the neurotrophic receptors for glutamate on astrocytes get activated, and several cascades generate these molecules that are controlling the blood supply. So astrocytes is a very important element of the synapse that controls energy supply in the brain. What's interesting is that in baseline conditions, neurons and astrocytes, they take up approximately the same amount of glucose. However, most of the energy is burned by neurons. 80% of energy in the brain goes into glutamatergic transmission and on generation of synaptic potential and action potentials. So, however, like astrocytes, they don't consume that much energy. It's only about 5 to 15% of energy that they consume. And for a while, there was a debate and a question where the rest of the energy goes. This was partially solved by the model that was suggested by Pellerin and Magistrati in 1994. That's astrocyte neuron lactate shuttle. Maurizio already presented this beautiful image. So it's a explanation that energy actually that is stored in astrocyte goes to neuron, but in another way, not in the shape of glucose, but rather in the shape of pyruvate and lactate. When glucose enters the lactate, there are enzymes that breaking it down to lactate and pyruvate. Then there are specific transporters on the membrane of astrocytes called the MCT that are extruding the lactate and pyruvate into extracellular space, and then those substrates are being taken by neurons by the other subtype of transporters for these substances, and then they are being utilized in neurons for energy. So inside of the neuron, lactate converts to pyruvate using the specific enzyme called lactate dehydrogenase type 1, LDH1. This is a process that also generates one molecule of NADH straight away. So it's kind of fast energy. The rest of pyruvate goes to mitochondria, and where they also convert it to energy, but this is a bit slower process as it takes time on different compartment, and the 3-carboxylic acid cycle takes also a bit more time. But these are the main energy cascades that are involved. And it had been shown that lactate shuttle is very important for memory and for expression of plasticity-related genes. A number of studies has been shown that plasticity-related genes like ARC, C-FOS, and ZIF are dependent on the lactate. So if you have an external lactate, you have higher expression of these genes. In this paper of Young, they also showed that lactate can directly amplify the currents via NMDA receptors that are involved in plasticity. So, and this happens as a result of generation of NADH molecule. As I said, when the enzyme converts lactate to pyruvate, there is an NADH molecule generated. And having enough NADH is important for activation of NMDA receptors. So that's what had been shown, that lactate is important for plasticity in these kind of studies. Then there was some behavioral evidences that lactate is important for memory, long-term memory. First, they show that if we block glycogenolysis in astrocytes using DAB, it's a special drug that does that, short-term memory would not be affected, but long-term memory would be completely impaired. And this could be reversed if the co-application of DAB and lactate could be done. So in this case, long-term memory can be restored. Similar effect is produced if transporters for pyruvate in lactate would be also blocked, either on astrocytes, either on neurons. Both of them would affect long-term memory. And what's interesting is that if transporters at astrocytic site blocked, but there is an external lactate, it could rescue long-term memory. But if it's lactate does not enter neuron, and if these transporters are blocked at the site of neuron, external lactate can no longer rescue the long-term memory. So it says that lactate entrance to the neuron is important. So that's what we studied in our project. Here we wanted to see like how and which energy substrates are allocated to different forms of memory engram. And mostly we wanted to focus on cellular level and used classical approach, studying synaptic plasticity in hippocampal slices. We used frat hippocampal slices and we recorded from C1 pyramidal neurons, stimulated Schaffer collaterals, and for studying plasticity and importance of lactate, we chose two protocols generating plasticity. Both these types are NMDA-dependent plasticities, which we verified afterwards. So, but we wanted to see how different patterns could be dependent on lactate presence. We chose like heavy plasticity, heavy protocol, in which we have 200% synaptic stimulation. So this is a theta burst. That's a classical approach to trigger plasticity in hippocampus. And we have this 5 theta burst. So it's relatively heavy load of stimuli. It's 200% synaptic stimuli. And then we chose a protocol which is spike time independent plasticity with much lower load. So it's only 50 stimulation at relatively lower frequency, 0.5 hertz. And both these protocols are able to trigger LTP in hippocampus. So this is just an example what kind of plasticity we get. So when we apply this protocol, the postsynaptic currents increase dramatically in both cases, in case of theta burst stimulation and in case of STDP. What we did next, we test whether this plasticity really depends on NMDA receptor activation. Now you can see that the black control line show that both these protocols successfully trigger LTP in hippocampal neurons. And if we do internal application of an NMDA receptor blocker, MK-AO1, we prevent this plasticity from occurring. So we see that both these plasticities indeed are NMDA dependent, and then we can test their need for lactate and glucose based on the activity pattern. And what we did next, we tested pharmacologically following steps, whether like to test how this astrocyte to neuron lactate transport involved in LTP induction. So first we tested whether glycogenolysis at the level of astrocytes is important. Then we tested the entry of lactate into neurons. Then we test whether the conversion of lactate into pyruvate is important for plasticity. And in the end, we checked the glucose and hexokinase involvement. So we start first with the glycogenolysis in astrocytes. For this, we also patched C1 pyramidal neurons, and we applied the blocker of glycogenolysis in astrocytes. This is showed in a green line. We see that in this case, we don't have plasticity, neither triggered by theta burst stimulation or CDP. Glycogenolysis is important for LTP induction in hippocampus, and we could rescue it if we would bath apply lactate. In this case, we start having back our LTP, so lactate would restore this function. Next, we tested whether lactate entry into neurons is important for LTP induction. For this, we blocked MCT2 transporters that transport lactate and pyruvate into neurons. And so by that, we prevented the entrance of lactate into specifically the recorded neuron, or we did it like only in the intercellular solution. So here we have our control shown in black, and we have our LTP in both cases, and theta burst and STDP. Here it's slightly different control because the drug is diluted in DMSO, so the control is also done with DMSO. In these conditions, we also have LTP shown in black, both. But when we block the transporters, we stop having our plasticity in both cases. Like there is no LTP, neither triggered by theta burst or by STDP. If we co-apply the transporter blocker and the lactate inside of the pipette solution, we were able to restore plasticity in both cases. So it shows that lactate entering to C1 neurons is necessary for LTP induction. Then we also tested whether it's specifically lactate or the other sources of energy that are important. So we did the same. We also blocked the transporters for lactate and pyruvate, and we co-applied it with pyruvate, and we see that actually pyruvate was not able to restore LTP. So it's not the pyruvate the key element for this energy, for these both plasticities. So then we tested like whether this molecule of an ADH that is usually formed by the enzyme that converts lactate to pyruvate is important, because as I said, it's a source of fast energy in the neuron. And when we co-apply the blocker of lactate transporters with an NADH directly to the pipette, we were able to restore plasticity. So overall, it says that it's entrance of lactate and the conversion is important. But then we move to the next step when we block the conversion of lactate into pyruvate. That is done usually by this lactate dehydrogenase 1 enzyme, and we used pharmacology also again applied into the patch solution. It's called oxamat. So when we patch a cell with oxamat, we interestingly lose our LTP in theta burst stimulation, but not in a CDP stimulation. So when we have a mild protocol that doesn't require so much of neuronal activation, lactate seems not to be crucial for this process. So, but when we have a heavy load of activity, as for theta burst, LTP is no longer here. So lactate is required for heavy load plasticity or 5 theta burst induced LTP, and it's not the case for CDP. Then we try to rescue this plasticity, whether like the presence of pyruvate would help, and we see that actually pyruvate did not rescue the LTP, so actually it's the lactate that is more important, and conversion of lactate into pyruvate with generation of NADH. Because when we co-applied oxamate and NADH into the pipette, we were able to restore LTP. So what also interestingly, we showed that it was earlier shown that lactate can increase NMDA receptor currents, but it happens only when lactate enters the neuron, then it can increase the currents. But if we prevent lactate from entering to the neuron and we have external accumulation of lactate, actually it produced the opposite results. The NMDA receptor-mediated currents, they inhibited, which is not the case for AMPA receptors. And this process also could have been slowed with the NADH present in the pipette. And if we co-apply, if we block the transporters for lactate and we co-apply it with NADH inside of the neuron, we can keep NMDA receptors current in a normal range. So this is aligned with the previous experiments of done by the group of Pierre Magistretti, showing that NADH from inside can stimulate NMDA receptors currents, but from outside it actually decreases them. So then to explore more the dynamic range and how these requirements of energy depend on different activity patterns, we used mathematical modeling. It was done by Hugues Perry, and he built a model that demonstrates that shows the energetic needs of C1 pyramidal neurons for plasticity. So this is like the scheme of the model. So we have several compartments. We have presynaptic neuron, we have postsynaptic neuron, and we have an astrocyte. Yeah. So the postsynaptic weight is modeled as bistable system gated by calcium and concentration of ATP, because ATP is important for LTP induction, and calcium triggers LTP. So when the calcium reaches certain threshold to trigger LTP, we have potentiation, and when we have consumption of ATP, so the ATP drops below a certain level, we have depotentiation. So basically, that's the way we model the postsynaptic weight. And for modeling the concentration of ATP, there are several processes that have been included, in particular glycolysis and activity of lactate dehydrogenase and astrocytes in postsynapse, and also the transfer of glucose inside of the neuron and lactate. So, this model well captured the observed patterns and kinetic of the plasticity that we observe with 5-theta burst stimulation and STDP plasticity. So as you can see here, and then we use this model to check how synaptic weight would change with this consumption of ATP with different protocols. So black line shows spike time independent plasticity triggered LTP, and the pink line is a theta burst induced plasticity. So in control conditions, when we have presence of all the energy substrates, we have elevated sodium in the neuron, so it has the activity. At the same time, lactate is consumed to support the activity, then a bit of glucose. And we see that actually in this stimulation, ATP level also drops, but not so much. So it doesn't reach the threshold, and we have generation of LTP. So we have change in synaptic weight, and there is a potentiation. It's the case for both STDP and theta burst stimulation. But when we block conversion of lactate into pyruvate, what's interesting here is that ATP concentrations start to fall below the level for ATP threshold, and we have failure of potentiation. So we see that actually in case of STDP here, we don't have such a strong decline of ATP level, while for theta burst stimulation, there is a lot of ATP is used for spiking activity, and as it falls below this threshold, we lose this potentiation. Then we use this model to make several predictions about the role of glucose and hexokinase. I should say that hexokinase is the enzyme that also starts the metabolism of glucose in the neuron. There is also important step of generation of NADH at this moment, not only when this LDH is active. And the model predicted that this effect of LDH, the presence of lactate, is important when the glucose concentration is physiological. I shall say that all these experiments been done at 5 millimolar of glucose, which is almost physiological, closer to this range, while many experiments in slices are done with much higher glucose, around 25. And what we see that actually here, if we increase the concentration of glucose, the inhibition of lactate will stop taking any effect. But when we have it in physiological range, we have this depletion of energy if we block lactate. So we tested it experimentally, and we did experiments of as before, but at higher concentration of glucose, particularly 25 millimolar. So we have control, and the same with oxamate. In both cases, we have plasticity. So in this case, indeed, the lactate doesn't play such an important role in generation of LTP. But I should say that 25 millimolar is very high concentration. It's not physiological. At physiological range, lactate seems to play an important role. And in this case, we just see that high glucose concentration bypass fueling of lactate. Then the model also, we wanted to see if glucose for this high energy plasticity would be important. And we see that in the model, hexokinase inhibition does not change the LTP induction. And we repeated this experiment, and we did this experiment, and we see that actually if we block hexokinase with the drug called monoheptulose, we don't lose our plasticity. Lactate is fully sufficient to trigger plasticity alone. Glucose is not necessary at this stage. But the lactate is crucial for this type of plasticity. Then we did similar predictions for STDP. And what we've seen that also hexokinase inhibition would not affect STDP. And when we blocked it in this condition, we didn't have any change. So lactate could still support STDP without having a big effect when we don't have glucose metabolism in the neuron. And then we tested the same for STDP when we had a bit more pairings and a bit more of activation. We also seen that the model predicted that hexokinase would not take any effect, and that's what we see also experimentally. We still have plasticity. And what's interesting that is when we co-apply the blocker of hexokinase and lactate conversion with oxamate, we don't have any plasticity anymore. So it seems that like STDP relies either on one pathway or the other, so it can be triggered via glycolysis or lactate pathway. So, but one of them should be present. Then we wanted to explore what kind of activity pattern would shift this need between glucose and lactate. So then we made a prediction for theta burst induced plasticity that if we have less theta bursts at some point, it would be sufficient to have only glucose, and lactate would stop taking any effect. So here we see that at 5 theta burst, we don't have LTP with inhibition of LDH, but if we have just 1 theta burst, we should observe LTP. And that's what we tested experimentally. And indeed, like when we have only one theta burst stimulation, it was not prevented, the LTP was not prevented by LDH inhibition. Next, we check the dependence of LTP expression when we do STDP. And we see that actually, no matter how many pairings at this low frequency rate there would be, lactate would never be so crucial for this plasticity. So here we increase just number of pairings. Now we have not 50 pairs at 0.5 hertz, but now we have 100 pairs at 1 hertz. And we see that it triggers LTP in control conditions, and in presence of oxamet, of LDH blocker, we still have LTP. So LTP expression would not depend so much on lactate. And then we ask the question when it would depend. So if it's the number of spikes, what is actually important? And what we found is it's actually if we increase the number of spikes per pair, it will shift our system from glucose reliance to lactate reliance. So here we kept the same total number of spikes, but we reduced number of pairings, and we had 2 backpropagating action potentials per each pair. And in this case, we found that oxamate in this type of STDP would be crucial to generate plasticity. So here we don't have LTP anymore when we have 2 spikes, postsynaptic spikes. And this could be reversed with an ADH. If we have an ADH, LTP is still here. So the conversion of lactate to pyruvate with generation of an ADH at this stage becomes important.
Yulia, you have about 8 minutes.
Okay, cool. Yeah. So similarly, like when we reduce the number of pairings, at some point this effect stops taking an action. And when we have only 25 pairings with 2 backpropagating action potentials, it stopped taking any effect. So we still have plasticity in this case. So this is how we checked all the activity pattern, and we showed the dependence of lactate or glucose fueling depends on exact number of spikes and frequency. And most crucially, that astrocyte-derived lactate is required for more energy-demanding types of plasticity as 5 theta burst-induced LTP. Then we moved to behavioral studies. We wanted to see whether this be also crucial for memory, and we used 2 behavioral tasks. One relatively easy for the animal, the other one would be more difficult. The easy one is the novel object recognition task, when there are 2 identical objects that animal would need to remember, and one would be replaced to another one on the next day. And we see whether the animal has the preference for the novelty. So we did it in rats, and we did it in control conditions, and in rats with the cannulas where we injected oxamate to inhibit LDH. And we see that normally in habituation phase, familiarization phase, animals like both objects. So the experiment worked. For the more complex task, we chose object in place task where we have 4 objects. And on the next day at the test session, we replace 2 of them, we change the position, so either one pair or the other. So, and here it's a more energy demanding task as an animal has to remember more objects, and it has to remember the position of the object. So it's, and we wanted to see if the block of LDH would also affect easy and let's say more difficult task in the same way. What we found is that actually when we test novel object recognition task, in case of saline, all animals show strong preference for novel object. And when we also apply oxamate, they still been able to show novelty. So this simple task seemed to be not dependent on lactate conversion. And the same we show with the preference index. Like all the animals like the novel object with even with the oxamate. But when we switch to object in place task that is more complicated, we see that the animals injected with saline were still able to recognize the novelty. They've seen that the objects were switched, while animals injected with oxamate didn't see this difference. So the main conclusion from this that conversion of lactate in pyruvate is crucial for more difficult behavioral tasks. Then we wanted to correlate this data with the plasticity that happens in hippocampus. So basically, we repeated the same experiment as the last one, behavioral, but we also recorded plasticity in the field of PSP in vivo in these animals. And we recorded it before familiarization and 2 hours after that. And we wanted to correlate this plasticity occurring with the ability of animals to learn the behavioral task. So it's the same task as I showed before. We switch the position of 2 objects and see whether the animals can see the novelty. What we found interesting is that in animals who were injected with saline, the animals who were able to distinguish novelty, most of them were having potentiation after 2 hours after familiarization phase. And animals injected with oxamate did not show that. Majority, although some had potentiation, most of them showed depression. So it seems like for challenging tasks, lactate is important for generation of LTP and recognizing the novelty. So here we're coming to conclusions and perspectives. So in this work, we show that 2 pools of energy substrates, glucose and lactate, are distantly allocated on demand, depending on the neuronal activity and the patterns. So lactate is mandatory for demanding neural computations, while glucose is sufficient for lighter forms of LTP, such as STDP. We also show that variations of spikes and frequency during LTP inductions are, even small variations are sufficient to change the fueling of neurons from glucose to lactate, like more energy demanding, more lactate is needed. And so we also show this behavioral task that lactate conversion into pyruvate is important for tasks where cognitive load is higher. And potentially this can be all important for the studies where energy fueling of the brain is compromised, such as neurodegeneration, Parkinson's disease, Alzheimer. So targeting specific parts of this pathway could probably restore the normal brain metabolism in these conditions and prevent neurodegeneration. And well, thank you for your attention, and thank you. I want to thank everyone who participated in this study. So Dr. Laurent Venance was my supervisor with whom we did this work. This work was done in collaboration with Pierre Magistrati. He was a great inspiration for studying this. Dr. Hugues Perry did the mathematical modeling, which really helped us to understand better the observed phenomenon. And Sylvie Perez and Charlotte Piet were my colleagues helping me with some surgeries and behavior. So that's it.
Thank you, Yulia. Very fascinating work. So we have time for a few questions. Please, I let the audience just type in the questions, and I proceed meanwhile to read the first question that I see here in the box. So actually, it's a double question, It comes from Milad Sultanzadeh, and he's asking, the first question is concerning your model. So first of all, does the model, if you know, include lactate from the blood circulation?
It does contain the lactate from extracellular space. So of course it would depend on the lactate supply from the blood vessels, but mostly I think it should be there. We can check the slide.
No, I
It
believe
could
you
have been,
didn't.
yeah, I think it's taken into account as an extracellular concentration of lactate.
And let me see. So accordingly, does he assume that only the astrocyte to neuron lactate shuttle exists, or is it neutral with respect to the direction of the lactate transport? Meaning, does he assume that the astrocyte to neuron lactate shuttle it's only from astrocyte to neuron, or should we also consider the fact that a neuron might shuttle lactate to astrocytes? That's a famous $1 million question in the
I think
ANLS,
we should consider that.
right?
Although, like direct measurements with fluorescent markers for lactate and pyruvate show that actually there is a gradient between blood vessels and astrocytes and neurons. So we expect that, like, usually in astrocytes it's much more than in neurons, and so we wouldn't expect that it would go the other way around. But maybe at local, like small places, like synaptic cleft, it could take an effect.
Okay. I see as questions hopefully come up. No, I see that there are no more questions here answered. So I will have a quick question and then wrap it up. So my understanding is that the demonstration that you have on STDP and TBS was in slices, right?
Yes.
Now, the question may be superfluous, but and the behavioral experiment actually support partially indeed the ANLS. But did you consider the possibility that at some point the income of glucose and therefore the blood circulation pathway could compete with the actual ANLS in vivo?
I guess it could.
What would you expect is the timing requirement for this kind of competition?
It could be a bit slower. It's like still most of the glucose is coming from astrocytes. So astrocytes provide a large proportion of energy to neurons.
Yes, the question comes from the fact that you're actually looking at glycogenolysis, right? Yeah. Which is a very specific source of this pyruvate, right? It's used in the moment in which we have very intense stimulation locally. But I mean, this is a remarkable, peculiar source of pyruvate for the cell. So in reality, like we have influx of glucose and glycolysis instead of glycogenolysis. So, yeah. So how would you combine these two things?
Well, glycogenolysis also passes through glycolysis process. So glycogen is broken down first to glucose, and then it goes through the same, basically, cascade. So
it's true, but glycogenolysis is something that we suppose the brain uses in very specific cases when it's short on energy, or it requires a very fast access to energy to learn something peculiar. And I mean, so it's I would argue that in a novel object recognition, as you did, glycogenolysis might not be as important as instead in survival conditions, or like survival tasks for learning something that is more dangerous for the survival of the species.
Yeah, I think we chose this task just to not to have some contamination from other possibilities. Like fear is a very strong process, very harsh behavioral task, and we wanted to have like cognitive task. And indeed, it was really tricky to find the proper one that would be showing scale, like cognitive load. Of course, we just assume that it's much harder for mouse to remember 4 objects and see how they move in space. But we do see that oxamate takes a strong effect in these tasks. So we assume that it has importance for this particular level of cognitive load. But it's really hard to correlate with the data that we get on slices.
Well, okay. Thank you very much, Yulia. That was fascinating results. And I would like to wrap up this session. I must owe an apology to all the audience for running late and literally devouring the discussion panel session that was supposed to follow my synaptic physiology session. But I owe to the great talks that we had to host during the session, and to the speakers and their effort to join despite some time constraints. So please accept my apologies, and I will forward the list of the questions that haven't been answered through email to the organizers in the next couple of days. And I see that the other organizers are joining me here, and so I personally thank you very much for the attention, and I hope you enjoyed the session, and I leave the word to the organizers to close up the session.
Thank you, everyone, and see you in the afternoon.
Thank you, everyone. Bye.
Thank you.